So hi everyone and welcome to hVIVO first ever LinkedIn audio event. My name is Egle Pavyde and I am Director Business Development for Europe and Asia at hVIVO. I will be the moderator of today's event and I will try to make it as valuable as possible for everyone online. We are happy to see that so many infectious and respiratory disease industry leaders are joining us today. And of course, if this event is successful and your feedback afterwards is positive, we will make sure that it will be a recurrent one on different topics around human challenge studies. I'm here in the room today with my two colleagues, so Bruno Speder and Nicolas Noulin, who are both experts in human challenge studies from very different angles, I would say. So I will now ask them to shortly introduce themselves, and Bruno, I will give a word to you to start.
Yes, happy to be here today. So I'm Bruno Speder. I'm the VP Regulatory Affairs. Been working in regulatory affairs for the past 15 years and have been helping a great number of infectious disease developers in their interactions with regulators on the use of challenge studies and bringing their developments forward.
Thank you, Bruno. Nicolas?
Hey everyone, my name is Nicolas Noulin. So it's almost 40 years I'm working for hVIVO now. I've got a PhD in molecular and cellular biology, where I used to work on toll-like receptor pathway, the innate immune system, often described as simple, but that we share because we share it with insects, and often underestimate in comparison to the acquired immune system, T cells, B cells, memory response. I also worked in the past at Pasteur Institute on H5N1 strategic for the vaccination. And as I mentioned, it's now almost 14 years I'm working for hVIVO, and my job currently is to lead the various GMP manufacture campaigns. Recently we have produced 3 SARS-CoV-2, Wuhan, the Delta, both have been used, Delta variant, both have been used in man. And recently we also produced the Omicron BA5 and some more recent flu virus such as H1N1 and B strain. And we just harvested an H3N2 yesterday. This virus was isolated in 2022, so quite busy, but we hope it will be a great session for you today.
So thank you, colleagues. Now I would like to shortly explain the concept of this event. So firstly, I will introduce you to hVIVO and our expertise in the field to give you just a bit of a background on our experience. Then we will move to a discussion part, which will be the main part of the event, where I will ask both Bruno and Nicolas to comment on the questions that we received from you before the event. You can also submit your questions in the comment section of the event, and if we have time, we will cover as much as possible during the event. If not, we will respond to them in written afterwards. So without further ado, I would like to give you some key info about hVIVO. So first of all, we consider ourselves a world leader in human challenge studies. And why do we think so? Because currently in the market we have the most human challenge models available. In addition, we have been conducting human challenge studies for more than 20 years now. And to date we have completed 75 studies and inoculated over 4,200 healthy volunteers with different challenge agents. We have a number of challenge models available. Some of them were already mentioned by Nicolas. So we work on different viruses and we constantly update our viruses. We are working on the new ones as we speak, and this is basically coming from the client demand on those. Our CHIM studies, so controlled human infection model studies, are conducted in our state-of-the-art facility in London. With a capacity of 50 beds, and we also have a Manchester screening facility to pull additional healthy volunteers from the northern part of the country to our site in London. So we support our clients from study design up to the clinical study report, all services included. hVIVO scientists leads the development of clinical study protocols, helping our clients to maximize the success of their studies. So besides the scientific expertise, our core strength is speedy healthy volunteer recruitment and a database of 300,000 of subjects that we currently have. So this was my very short pitch just to give you an understanding about hVIVO. And now I would like to give a stage, of course, for our expert to explain you more about human challenge studies and what are the benefits of including them in your drug or vaccine development plan. So, Bruno, the first question is for you, and I will be quite straightforward with this one. So, why would I, as a sponsor, conduct a human challenge study from the regulatory point of view if this type of study is not mandatory by any regulatory guidelines?
Yeah, and it's actually an excellent question. And when you look at human challenge trials are like any other trials. And when we do clinical research, our aim is to collect data. In your whole clinical development, your aim is to collect data to bring your product to the market. And a human challenge trial is actually an excellent way to, depending on the stage you are in development, to have a very quick and early view on the proof of concept, efficacy-wise, of your vaccine or antiviral. And of course, the endpoints depend if you're testing a vaccine, an antiviral, the disease, etc. It's always a bit of case by case. But human challenge trial is, like any trial, a tool to collect data. And the data you collect in a human challenge trial will allow you to make certain decisions, and mostly to have a first insight in the efficacy, but also to help you in designing the further stages of development to further guide your development pathway. So yes, when it's correct that these are not mandatory by guidelines, these are a very useful tool to further advance your development.
Thanks, Bruno. So, Nicolas, on the same point, what are the main reasons why pharma or biotech companies decide to conduct human challenge studies, and maybe more from a scientific point of view?
Mm-hmm. So, one key aspect of the human challenge trial is that it's a controlled environment where we know the challenge agent, we know the dose we give to the volunteer, we know how we give this dose, and by the way, just on the side, the dose for a vaccine efficacy or a drug efficacy may not be exactly the same. So we can adjust the dose of the inoculum. We know when we give the inoculum, and more importantly, or quite importantly, we have pre-challenge baseline from all our volunteers. So the baseline are key to this kind of trial because you can take blood samples, swab, you can analyze this data, the baseline data. These are key data that you would not get so easily in a field trial. So overall, pharma and biotech, they come to us with the aim of accelerating their development. It can also be candidate selection. I've got 2 vaccines. I would like to select one. Which one should I go for? So you can eliminate your candidate before they cost you too much money. So you can accelerate and eliminate the candidates that are too weak. And most of the time, the result leads to rapid modification of the vaccine or the drug candidate and to dose adjustment. So doing this in the field, we really feel, and we have data that it takes years to do those. So just to summarize for this question, I think for the big pharma, it does support their internal decision-making process and prioritization. And for the small biotech, most often it's financially linked. It helps them raising more money, further capital. And it takes their lead candidate to phase B or phase 3. It's a much different expertise. It's not at all the same expertise. Small biotech, we speak 10, 20, 50 people working together on a project, and bringing this to phase 3, it's a completely different expertise.
Mm-hmm. Thank you, Nicolas. Bruno, I know you have supported many vaccine and antiviral drug developers in their interactions with regulatory bodies. So what is the perception from regulators on these type of studies, and is it more positive or negative?
Well, the data from human challenge studies have been perfectly accepted by regulators on both sides of the Atlantic, both in Europe as in the US. And when you see in interactions with regulators to include a challenge trial in a development program, be it for an antiviral, a vaccine, sometimes even immunomodulatory product, it is certainly a concept that regulators are very open to discuss. Of course, when you look at the data collected, and it comes back a bit to the first question, is clinical research is all about data. So, and regulators look at what data do they want to see to bring your product either to the next phase of development or to the market. And where challenge studies can help, regulators have always been very open to consider these data and to include them in the development pathway of products. So in general, these data have always been very well received. Of course, this always comes with the caveat that no product is the same, no disease is the same. So it's always a very case-by-case approach with a very tailored development. But in general, these data have been very well received.
Yeah, and Bruno, you mentioned different authorities. So, for example, if we are taking the two biggest ones that we usually work with, EMA and FDA, so how they see human challenge studies, and are there any differences in views from EMA and FDA?
Yeah, well, it's a very good question. When you look at studies, there's always a little bit of difference between the FDA and EMA, is that the FDA, of course, is both in the US the agency that approves the products on the market and approves clinical studies. Where, of course, in Europe we still have this setting, even with the new clinical trial regulation, where the studies itself are approved by a national regulator. And like always in infectious diseases, EMA approves the marketing authorization to the centralized procedure. So in Europe it's always a bit this interplay like you always have between national regulators and EMA. But in general, both EMA and FDA have been quite favorable to human challenge studies. Of course, when it comes to the running of the studies itself, there is always a bit the different view on the risk-benefit, which is also quite culturally linked, and that's always I think something that we tend to forget in drug development is that risk-benefit assessment and what is risk and what is a benefit. The views sometimes differ on both sides of the Atlantic. It's a bit culturally driven. But in general, both agencies accept these data, and both agencies are open to have these studies conducted. So I would not say that there are real differences, but of course, if you look at specific indications, there's always that risk-benefit approach that is a bit different on both sides of the Atlantic.
Okay, thanks. That's clear.
Can I just ask Bruno about the MHRA? As we are in the UK, we are regulated by the MHRA. How would you see MHRA position closer to EMA or FDA or?
Well, that's a good question. So, of course, since Brexit, the MHRA has left the EU regulatory system, where the MHRA is now a standalone sovereign regulator, a bit like the FDA is for the US. If you look at the MHRA, the way the MHRA looks at studies and the way the MHRA looks at its place in the development of products it is still quite like it would have been in the EU regulatory system. The MHRA has, of course, an historical knowledge and historical expertise in challenge studies, which allows specifically on the study field and the tactical fields to have studies approved earlier, and they are quite let's say, they have a more risk-benefit approach, and they have a more approach to, they're more open for discussions. I think that the COVID characterization study and discussions around that illustrate that. But I would say in general the MHRA is quite still in line a bit with the European thinking. Now, of course, in the coming years we will see how the MHRA evaluates in its thinking, as it's now an independent regulator. But let's say the first signals we get is that the MHRA is still there to, let's say, advance science, advance research. A number of initiatives have been taken in that. So I think that the UK will still be a good place to run this type of studies today and in the future.
Okay.
Yeah, if I may, just one last point is that not this kind of human challenge studies are not allowed everywhere in the world. So, for instance, I come from France, where it's not allowed to run such a study. So there is really some kind of context in this research.
Yeah, so thanks, Nicolas, and now the question comes to you. So are there any limitations in terms of challenge agents? So can human challenge studies be conducted basically on any infectious disease?
Well, I would directly say no. We don't want to inoculate volunteers with any disease. But one thing is for sure is that all studies that are run are ethically approved. So, and you know, the clinical protocol is peer-reviewed. Usually you involve key opinion leaders in the writing and the setting up of such a study. Safety is and will stay first priority for us and for everyone running such a study. And when a new challenge agent is used for the first time in man, we and others are doing titration study, in which we usually do dose escalating or de-escalating dose finding. It can be unblinded study or single-blinded. Also, just with regard to the challenge agent itself, there are two approaches. We have viruses, for instance, such as the one we run. We try to run with the virus that is as wide as possible. Basically, there is no mutation induced by us. For instance, mutations that would attenuate the virus or the pathogen. But for some pathogen, you have to attenuate the pathogen. You cannot, for instance, inoculate tuberculosis, hence why some researchers are using another mycoplasma, another pathogen, to mimic as close as possible to the virus or the pathogen of interest. And you can also target specific populations. The screening of the volunteer is key. For instance, there has been a recent clinical trial involving Zika. It's very recent, like this was published a month or two ago. And on that trial, they only use female volunteer. Okay, so the screening, the population you target, and the pathogen you're using are driving whether or not you should do your clinical trial. But to answer your question, no, you cannot do with any pathogen.
Okay, thanks, Nicolas. So, Bruno, a very specific question comes to you on the cholera vaccine case. So, this vaccine obtained a marketing authorization straight after conducting a human challenge study. And can this vaccine registration pathway be used for any other indication?
Yeah, that's a good question. And maybe for the people listening that are not very familiar with this particular case, so this is the Vaxchora cholera travelers vaccine. So it's a vaccine, the marketing authorization holder is PacVax. I think the marketing authorization was recently transferred to Bavarian Nordic for a number of countries. So it's a cholera vaccine that's on the market, both in the US and in Europe, for cholera, prevention of cholera, but specifically for travelers. And in the development plan, or in the data presented to the regulator, the cholera challenge study was used as the pivotal efficacy element in the package. Of course, there was still a big phase 3 study done, but more for safety and immunogenicity to build the safety database, as you'd always do with vaccines and with any other drug, but with vaccines you usually have a somewhat larger safety database. But the human challenge study was used as pivotal element to bring the product on the market. And is this a principle that can be used for another indication? In principle, yes. I would say in principle, yes. Of course, there are a few caveats with this. Is of course, in this particular case, the population in the human challenge study, young, healthy people, are also the population that's targeted for once the product was on the market. So the population in the challenge study mimicked the travelers population that would get the vaccine. Also important to note is that there was already quite some extensive data available on a very similar vaccine, very similar cholera vaccine, and that made the clinical development relatively short because there was no dose selection, etc. to be done. So that's also an important element that plays. But in principle, would it be possible to bring other travelers vaccine in a tropical disease, for example, to the market using this approach? In principle, yes. Of course, as a regulatory person, this always comes with the caveat. Should regulators approve? It depends on the disease. And for example, in dengue, where you have the risk of reinfection, etc., that might be a bit more difficult. But there could be other diseases where that could certainly be an option that could be considered, certainly.
Could you name an indication like that?
I think, for example, typhoid or another bacterial disease could be a possibility. I've had, but that's already a few years ago. But these discussions were still quite conceptual. Discussion on a yellow fever vaccine. Of course, with yellow fever, there's a bit of debate: can you run a challenge study or not? But these type of diseases could be used. Of course, and that's also maybe. The underlying question is, should you develop it as a traveler's vaccine? These data can, of course, always be used to, in a second stage, also develop it more as a vaccine for a population living in an endemic region. So you will certainly be able to piggyback on part of the data package. So it could also be seen as a kind of two-step development. First, you develop it for a traveler's population. And then in the second stage, you develop it for an endemic, for a population in endemic region. That's certainly a possibility. But as I said, I think already a few times in this call, it depends on the product indication, but also on how regulators would look at it. But in summary, in principle, yes, it can be used for a similar approach.
Okay. Nicolas, a very opposite question to you. So are there any cases when you would not recommend for a sponsor to run a human challenge study, and what would be the reasoning behind? So a sponsor comes to you, they have a drug or a vaccine, and you say, well, sorry, no, it's not for you.
Well, this has not yet happened, I have to say. But as for any scientific experiment, the risk-benefit must be known and evaluated. This meets what Bruno was telling us, the risk-based approach. This is a risk-benefit. And of course, you have to involve key opinion leaders, as I mentioned already. You can ask the question to the regulatory agency whether they think that it is a good thing and what it would add to your program. You have to have a clear purpose for doing such a study, and the model should have been shown to be safe. Okay, so if the drug or the candidate vaccine can exacerbate the pathology induced, then I would be careful and ask my clients or partner to think twice. But usually, where we have some kind of debate with clients or with the company we work with, is more on the N number, meaning we want to avoid the clinical trial to be underpowered. Okay, that is, if someone wants to test 5 doses of their vaccine or of their IMP, we say less than 15 or 20 subject per doses, then better test only 3, but increase the N number so that you have a better visibility on the outcome of your trial. So that's what I would suggest, is make sure that your powering is strong enough using the data produced during the characterization of the challenge agent.
Okay. And Bruno, we got a question from at least 5 registrants to this event, and this is also the usual that I get from clients, especially those who are new to human challenge studies. So is it ethical to run human challenge studies? And could you comment on that, and what are the differences from the informed consent perspective? So comparing human challenge studies and regular field studies.
Yeah. Well, it's a good question. It links a bit in somewhat in the previous questions. Is the principle of challenge studies ethically feasible? Yes, I think. Otherwise, you wouldn't be here today. But of course, this depends on the disease. This depends on the indication, but also on the population you include. And also there is a very culturally driven approach. For example, there are a number of diseases, let's say more parasitological diseases, where you would say, would it be ethical to run these studies in Europe? Well, the answer would be no, because we would say, yeah, but the disease doesn't occur here, etc., etc. Where, for example, in the endemic regions, where there is actual the disease and where people see the devastation certain of these diseases cause, because disease is always something, as long as you're not confronted with it, it's a bit far from your bed. That there, the cultural perception is, yes, we can run these studies, because for us it's important that drugs, vaccines are developed. So that ethical aspect is something also that is also a bit culturally driven. Would I, for example, run an influenza challenge study in Africa? Probably not. While here in Europe, we find that very normal, we run influenza challenge studies, and vice versa. So, is it ethical to run these studies? Yes. There is the cultural perception, there is the endemic aspect. You run certain studies in certain regions, because that's where the disease occurs. To come to the second part of the question, is the informed consent process different than in a regular study? No.
Is it more extensive?
Well, actually not. Of course, you explain the risk. It's always very important and critical. It's one of the bases of GCP is that the subject is fully informed about the risk, fully informed about the benefits, which in challenge studies for subjects is relatively limited to non-existent. But the process itself is the same. And when people say, look, there is a higher risk with challenge studies, that's not per se true. And for example, when you go to, for example, first in human studies, you have the same ICF process, and you know much less about the product. Specifically with certain therapies, there could be a risk. Also, there is no benefit for the subjects. So that ICF process is actually very similar for a challenge study than you would have for a phase 1 healthy volunteer standard safety study. So that would be a very similar process than, for example, to a field study where there might be more of a benefit for the patient or the subject participating in the study.
Yeah, that's clear. Thank you. Nicolas, so talking about the data obtained from human challenge studies, is there a difference between field studies and challenge studies, and what are those differences?
Sure. So, as I mentioned earlier on, it's you controlled most of the parameters, and thanks to this, you obtain clean data faster. So this is one of the main benefits of challenge trial, is that you have your clean data faster, and usually they are done in one site only. So, for instance, in our quarantine we have one site or possibly two sites, but very close to each other. So you don't have a multi-site field study with sometimes up to 50 sites in the world. You don't have to follow the challenge agent, the pathogen, say north hemisphere during winter in north hemisphere and south hemisphere during the winter over there. So you have your laboratory that is just a few meters from the quarantine area. So meaning that, say for instance, you want to stabilize your samples fast, then within minutes they go from the patient or from the volunteers to the lab, and then they are stored in aliquoted and stored in freezer. So this is giving you really clean sample, and we know now that we are no longer targeting one cytokine or one biomarker. You target many. You do biased or unbiased approach in your data and sample analysis. So having clean sample and clean data faster is really what make the challenge trial much more efficient in getting data fast. So, and just to say, in this, there are two type of challenge study, a big type. One is as inpatient, and the other is as outpatient. But still, when it's outpatient, you have direct contact with your volunteer. You have a medical team that is always looking for the volunteer. So, and you have a schedule of event or schedule of assessment that is thoroughly followed by the team. Everybody is trained. Everybody is working together to make to avoid the deviation. Deviation, you have far less deviation in such a trial than in field study. So that's
And Nicolas, another question to you on the basic perception, I would say, to human challenge studies, and we also got a couple of questions on this regard. So it is thought, at least, that human challenge studies are much more useful for vaccines rather than antivirals or immune modulators. So what is your view on that?
Well, it is true that we could see this as easier. It is easier to run because you screen your volunteer, then you invite them for a vaccination, for 1 or 2 visits, and then usually a month after you invite them for the challenge. So it is leaner as in the schedule of event. So vaccination, follow-up visit sometime to take sample, and then challenge. So that's, but on the other side, we have done, and others have done, many drug testing studies, and now we have tools. For instance, if, say, I'm inoculating a volunteer at midday on Monday, and then at midday on Tuesday, I can take a swab or a sample, do a rapid diagnosis, either a lateral flow device or by PCR, and when a volunteer turns positive for the pathogen, then we can dose this person with the drug that we want to test. So thanks to the modernization of the diagnosis toolbox, we can run, we and others can run drug study in exactly the same way as when you test a vaccine study. One last point about this is it's about the N number again, the powering, because when you run a vaccine study, you have to include your N number have to be slightly higher than when you run a drug testing study. Say, because a drug we dose when a volunteer is positive. Okay, so that's your N number is the number of volunteers that turn positive. Whereas with the vaccine, well, you have to inoculate more and vaccinate more. So, but the two of them now are quite easy to run. Yeah.
Bruno, can you comment shortly on what is needed from the clinical trial application submission package perspective? And again, are there differences between CHIM studies and trial regular field studies?
Yeah. No. The only difference is, of course, and that depends a bit from, let's say, country to country where you run the study, you would submit documentation on the challenge agent itself, which usually would be some CMC information and then clinical, non-clinical information, reference safety information. But that would be the only difference. And exactly which documentation you submit depends from country to country. In the US it's a little bit more extensive than, for example, in the UK. But in principle, the CTA package, CTA process is exactly the same. It's just that you add some documentation on the challenge agent you would use, and it would be assessed by the regulator reviewing the study.
Okay. We got a couple of questions, in the same way, so I will direct it to both of you, because I think it's very important. So how human challenge studies translates into field, especially when the target population is not healthy volunteers for that drug or vaccine between 18 to 55 years old. And the example was written by one of our registrants, so for example RSV. The target population is either premature newborns or elderly, so it's basically the opposite that the CHIM study population will include. So how does it translate, and are there any good examples showing this translation from human challenge study into field studies? So Bruno, maybe let's start from your side.
Yeah. It's a very good question, and it's actually a question with different subsets, and I leave to Nicolas to discuss more.
Yeah, she always give you,
but it's indeed correct that, for example, in RSV, you would run a challenge study 18 to 55 years old. That's a bit a typical age you would run these studies, and of course the big question is, are you developing a vaccine or a more therapeutic product? Now let's take a vaccine a bit as case study here. Yes, indeed the population in the challenge study is different than the population you would use, or that would be the target for your vaccine in development, and also the population in the pivotal phase 3 study. But what you see is, in this case, you're going to challenge study as a proof of concept study. And like in every proof of concept study, you want clean data. Do I think my product could work or not? That's basically the question you answer in a proof of concept study. And in that case, yes, a challenge study is perfectly fine to de-risk that program and to move the program forward. And that's, for example, an approach that Pfizer took when developing their RSV vaccine that has been received marketing authorization in the US, EU earlier this year. They ran an RSV challenge study in a population 18 to 50 years old. Analyzed the data, look, based on this challenge study, our vaccine seems to have an efficacy signal, and that allowed them to move to phase 3 in a population of elderly, and then for the maternal vaccine to the maternal study. But that's an interesting example where in a regulatory development, a challenge study has really allowed to move a product forward. And yes, of course, it's not the population you would really see in your phase 3 study, but that's not that different than in other therapeutic areas, where in your phase 2 study you have a more restricted population, often a somewhat healthier population. So the basic concept is the same, that you have a clear population and then you move forward to the phase 3 study.
Mm-hmm. Nicolas, any comments from your side on this one?
Yeah, just to complement what Bruno stated, I mean, the FDA recently made some clear statement recommending challenge study to be considered for such a product, for RSV, for instance. And of course, we will not challenge anyone less than 18 years old. That's not the aim. But we have done, and some others have done, challenging older volunteer, older than 55 years, up to 70 plus years with RSV. May not be the best to check efficacy, but at least you will understand how the pathogen react with the body of someone that is older than the 18 to 55 years old usual population that enters such a trial. One key aspect as well is not RSV specifically, but for flu. The challenge volunteer, you can test if the current vaccine is working against the current strain circulating. So you can pretty much have live indication before the season is over. You can tell whether a vaccine will work, and that is key, I think, for the taxpayer and also for the industry, especially for flu.
Okay, thank you. We have one very quick question coming from our registrant. So, is COVID-19 human challenge studies validated, and how do you validate new variants? So I think I can quickly comment on this one. So yeah, I think Nicolas already mentioned the different variants we have. Currently, the most recent one is BA5 strain. It's already GMP manufactured and ready for characterization. And of course, as with any other virus that we develop, if there is a client need to develop a new strain, a new variant, we for sure are ready to talk about it and to discuss further, because we always follow our client needs. We have only 2 minutes left, so I think I will wrap up here. So I would like to thank both Bruno and Nicolas for being with me here today, for sharing their knowledge. We still have some questions which we haven't managed to answer. We will make sure to post all of those responses in written to those to whom we haven't answered to your questions. And I will contact you to get your feedback. Did you like the event? Was it useful? And if so, if it will be positive, we would like to continue even going more specific with the topics on the next events that we would host. So thank you everyone very much for being here today, and we are very happy to see that so many of you joined online. And have a nice day. Hope this was useful.
Thank you, Egle.
Thank you very much.
Thank you, everyone.
Thank you. Bye.